In this video, I want to talk a bit about the microanatomy of the heart, the cardiac myocytes. Myo means muscle, and cyte, of course, means cell, so we're talking about the cardiac muscle cells, also known as the cardiac muscle fibers. You may want to review the structure of smooth muscle, and particularly skeletal muscle. We'll mostly talk about how cardiac muscle microanatomy is different from skeletal muscle. This is showing you a view of cardiac muscle under the microscope. Note how it is striated like skeletal muscle. You can't see the striations everywhere, but there are some areas where they're pretty clear, that sort of thumbprint appearance. There, in there, there. But cardiomyocytes are different from skeletal muscle under the microscope in a couple of distinctive ways. First off, the muscle fibers can vary in diameter and branch. There's a branch point right there. If you aren't quite seeing it, it's highlighted over here. Probably the easiest way to tell the difference between the two is the presence of these intercalated discs. They are perpendicular to the long axis of the cell, like the striations, but they're very dark. And these intercalated discs, they mark the boundary between neighboring cardiomyocytes. Here's a side-by-side comparison. Skeletal muscle on the right, you can see very clear striations, and very long cylindrical muscle fibers. Cardiac muscle on the left. Clear striations and intercalated discs there, there, there. Intercalated discs hold cardiac myocytes tightly together. You find two kinds of specialized cell junctions here. Gap junctions are little protein channels that allow ions to pass directly from cell to cell. So ions can basically, in one cell, can diffuse straight through the gap junction into a neighboring cell. So these guys allow direct communication between the cardiomyocytes. This direct communication allows the myocytes to synchronize contraction so that your heart works as a coordinated unit. Second type of junction are the desmosomes. Desmosomes mechanically anchor the cardiomyocytes to each other so that your heart doesn't rip apart during contractions. And I'm sure we can all agree that that is a very good thing. The desmosomes act a little bit like rivets. You have proteins that join together, half from one cell, half from the other cell, and they're anchored to the cytoskeleton in each cell, so that force of contraction is spread out across the entire muscle fiber. I just want to briefly remind you about the membranous structures that help out with contraction of skeletal muscle fibers. You have T-tubules, that's these structures in white here. T-tubules are infoldings of the plasma membrane that carry the action potential deep into the muscle fiber. So when that muscle fiber is stimulated, the action potential races down the T-tubules. Next to the T-tubules, actually one on either side of each T-tubule, you have the terminal cisternae. And the terminal cisternae are modified smooth endoplasmic reticulum. They're basically bags of calcium just waiting to be dumped into the cell to begin contraction. So when skeletal muscle is stimulated, action potential races down the T-tubules, opening calcium channels in the terminal cisternae, leading to calcium pouring out of the SR into the cytoplasm and activating the cross-bridge cycle. In the cardiac muscle fiber, you do see branching of cells, as we mentioned in the microscope image. The sarcoplasmic reticulum in blue looks a little bit different too. There are no terminal cisternae. There are also fewer T-tubules. Much of the calcium in cardiac muscle stimulation comes from outside the cell, from the extracellular fluid, so it's not as necessary to have the big bags of calcium ions around the T-tubules. One other thing to note is that cardiac muscle cells are very densely packed with mitochondria. This means that the cardiac muscle fibers are very fatigue resistant. Again, a very good thing. You certainly don't want your heart to become tired and unable to contract. You do see the regular thick and thin filaments, the thick filaments and thin filaments, arranged into sarcolemmas and myofibrils, just as you see in skeletal muscle. And remember, it's this arrangement of the thick and thin filaments that leads to the striations. So since you see striations in both cardiac muscle and skeletal muscle, you'd expect to see this regular arrangement in both. Alright, this is a short video, but it serves as a good introduction to the microanatomy of the cardiac muscle cells. So after studying it, you should be able to compare and contrast cardiac myocytes and skeletal muscle fibers, talk about the gap junctions and desmosomes that you see at the intercalated discs, and distinguish between cardiac muscle and skeletal muscle under the microscope.